Hello everyone, welcome to this segment. We'll be discussing about the specimens that have been provided by Waec for agricultural science practicals for the year 2024. I would encourage you to pay attention and also invite others by sharing this video to as many students who will be writing or sitting for this examination. Now I want you to understand that agricultural science examinations covers three aspects. We have paper 1, paper 2, and paper 3. Paper 1 has to do with objectives, which is about 50 questions. Paper 2 has to do with theory, which is about 6 questions, of which you are expected to answer 5. And paper 3 has to do with the practical aspect. And here you have 4 questions, and it will take about 1 hour, 30 minutes for you to answer those questions. And you will be, these marks accrued or awarded for this segment is 60 marks. So we're going to discuss in details the various specimens that have been provided by Waec. Pay good attention and follow me as we proceed. Now the first specimen here we have is called the ranging pole. Now that's a picture of the ranging pole. So let's look at the description of the ranging pole. You could be asked to describe the ranging pole. You could be asked to state the functions of the ranging pole. You could also be asked to state or explain the maintenance of the ranging pole. So starting with the description, let's look at how to describe the ranging pole, which is our specimen A. Now the ranging pole is made of wood or light metal. Now, it could be in various lengths, about 1.8 meters, 2.4 meters, or 3 meters. It's usually circular or round. It's also painted black, white, orange, or red, and this is to enable one to see it from a distance. Now, these painted divisions are in a specific measurement, and the ranging pole also has a pointed metal end. So this is how you should describe the ranging pole. Now let's look at the functions or the uses of the ranging pole. Remember, the ranging pole is specimen A. Now what is it used for? It's used for marking survey stations. That has to do with points. So when you're taking your measurements, the ranging pole helps you to get those points, which are called marking survey stations. So the second use is that it helps to measure offset. And what does offset mean? It means short distance. So the ranging pole can be used to measure short distances. And it can also be used for making straight lines over long distances. Now, how can we maintain the ranging pole? First, you should oil the metal part after use to prevent rusting. Next, you should paint the bands when necessary. You should also sharpen the pointed end if blunt, and you should also store it in a cool, dry place. The next specimen we have is the arrow. Now, this is specimen B. The arrow is also called the survey pin. Now, let's look at how to describe the arrow. Now, this is a thin pointed steel wire of about 30 centimeters long with one end curved into a ring. So from the picture, you can see how the description goes. It tallies with what I'm talking about. And the second thing you should mention while describing it is that a red cloth is usually attached to the ring so that it can be seen from afar. Now, what's the use of the arrow or the survey pin? It's used to make chain lengths and is also used to make survey stations, which is the same as points. Let's look at how we can maintain the arrow or the survey pin. First, you should sharpen the pointed end when blunt. Next, you should wipe with oil or grease after use when storing. You should also store it in a cool, dry place, and finally, you should clean it after use. The next specimen we have is the measuring tape, and this is specimen C. Now, let's describe the measuring tape. Remember, you could be asked to describe the measuring tape. Now, it is usually made of linen or fine steel sheet. It is usually marked on one side with the metric units and on the other side with the imperial units, and is used for various types and lengths. Okay? Now, it's normally wound in a small case from where it is unwound for use. So this is how you should describe the measuring tape. Now, what is the measuring tape used for? Remember, the measuring tape is specimen C. Now, what is it used for? It's used for taking measurements of length, breadth, and height. It's also used for taking offset triangles, and it's also used for determining straight lines and right angles. How can the measuring tape be maintained? You need to clean and dry it after use, or you wash and dry. You should also roll it back into the case after use. You should wipe the steel tape with oil before storage, and you should also keep it in a cool, dry place. The wooden peg is another specimen we're going to have a good look at. Now, the wooden peg is specimen D, and let's look at how to describe the wooden peg. Now, they are made of stout timber, about 5 centimeters square or circular size, and about 40 to 60 centimeters long. They are tapered at one end. What does that mean? It has a pointed end, so that they can be driven into the ground with a hammer. What is the function or the use of the wooden peg? It is used to mark stations, which has to do with marking points, and they are kept at about 40 centimeters minimum, projecting above the ground. So the picture helps to give us an explanation of how the wooden peg should be when placed in the ground. Now let's look at how we can maintain the wooden peg. First, you should choose high quality wooden pegs made of durable wood such as oak or teak. You should also apply protective coating to the wooden pegs to prevent termite attack. And finally, you should store it in a cool, dry place. At this moment, we'll be looking at an experiment on capillary action. So the title of this experiment is The Determination of Capillary Action, and the aim of this experiment is to compare the capillary action of different types of soil. So we have some apparatuses that will help us to conduct this experiment. Here we have 3 retort stands, we have 3 beakers, we have the capillary tube, we also have funnels, we have the glass rod and the pestle, or the glass mortar and pestle, we have the cotton. So all of these plus the 3 soil samples, which are the sandy soil, the clay soil, and the loamy soil. So all of this will help us to conduct this experiment. And the first thing we're to do is to grind the loamy soil. So this is to get rid of lumps in the loamy soil. Okay, so I would stock the cotton wool at the base or one of the open ends of the capillary tube. And I will now have to put the loamy soil into the capillary tube. So I have to make use of a funnel to achieve that. Okay, so I will now use the retort stand to hold the capillary tube. So I have to do this for the 3 soil samples. So now I have to also grind the clay soil. So the purpose of grinding the clay soil is to get rid of lumps. So next, I'll stock cotton wool at the base of the capillary tube, just as I did to the loamy soil. And I now fill the clay soil into the capillary tube. Okay, so we are supposed to tap the capillary tube to get rid of air spaces. Tap the capillary tube to get rid of air spaces. So I should also do that for the loamy soil. Now the sandy soil is not going to be ground, so what was recommended that the clay soil and the loamy soil should be ground. So I've done that and I've put them in the capillary tube, and I've also supported them using a retort stand. So next I'm going to put the sandy soil into the capillary tube. Okay, so I'm stocking the cotton wool again for the sandy soil, the capillary tube that will have the sandy soil. So I have to stock the base or one open end of the capillary tube. And I have to, so I'm supposed to fill, next is to fill the soil sample into the capillary tube using the funnel. Okay, so I'll now also support this capillary tube containing the sandy soil using the retort stand. Okay, so at this moment, now that we have put the various soil samples into the capillary tube, following the proper procedure of stocking the capillary tube with the cotton wool and putting the various soil samples, tapping the capillary tube to ensure that there are no air spaces within the soil samples, and suspending it inside the trough, water trough or beakers. So at this moment, I'm going to pour in water and watch and observe how the water will rise in the various soil samples. We'll observe how the water rises in the sandy soil, how it also rises in the clay soil, and how it also rises in the loamy soil. And when we observe that, we'll now move on to the next step of this experiment. So at this moment, let's pour the water. Okay, so it is observed that the water rises faster in the sandy soil as compared to the clay soil and the loamy soil. So at the initial stage, the water rises faster. And remember, capillarity action has to do with the ability of water to rise in a soil sample. So at the initial stage, the water rises faster in the sandy soil from observation. But remember, this experiment is to last for 24 hours. And it is also expected by the end of this experiment that the rate of capillary action in the clay soil and the loamy soil will supersede that of the sandy soil. So initially, the rate of capillary action is faster in the sandy soil, but at the end of the experiment, which is 24 hours, the rate of capillary action in the clay soil will be highest, followed by that of the loamy soil. So now we're going to look further at various possible questions that can be asked concerning this experiment. Okay, so we're going to look at all we need to know concerning this experiment on capillary action. So we need to know the title of the experiment that was just conducted. So the title is going to be Determination of Capillarity Action of Different Types of Soils. And the aim of the experiment is to compare or determine and measure the capillarity or rise in water of different types of soils. I mentioned all of these while I was demonstrating the experiment. Now let's take a look at let's discuss the observation or what we observed while conducting or demonstrating the experiment. So remember, when I was demonstrating it, I said there were certain expectations or what actually took place at the initial stage and what is expected at the end of the experiment, which is after 24 hours. So this observation is going to be based on what is expected at the end of the experiment after 24 hours. So here we have the clay to be the highest in terms of capillary action. Remember, capillarity has to do with the ability of water to rise up in a medium, in a soil sample, in a medium. So here, specimen G, which is loam or loamy soil, is higher. Thus, we describe it as being higher. And for the specimen E, which is sandy soil, will be the lowest. So the level at which water rises in the various soil samples, for clay soil, it will be the highest, followed by loamy soil, which will term higher, and for the sandy soil, we describe it as the lowest. Now let's also look at the apparatus that were used. Remember, I described or I mentioned the various apparatuses that were used, so we also need to know that you could be asked to mention the various apparatus that were used during the experiment. So we have a large beaker or basin or dish or trough, 3 capillary tubes or glass tubes, as I mentioned while demonstrating, 3 soil samples, which is labeled E, F, and G, in the capillary, that's on the various capillary tubes. These various soil samples were labeled E, F, and G. Water was also provided, cotton wool, and also the retort stands, which are also called clamps. Now, what method was used? You could be asked to describe the method that was used to carry out this experiment. So let's take a look at how you are going to present the method used. So first you say, take 3 glass tubes and plug each with cotton wool. Next, you say fill each tube with dry samples of clayey, loamy, and sandy soils respectively. Next, you should fix the 3 tubes in an upright position with clamps or the retort stands as demonstrated in the experiment. The next point you say is get the 3 glass tubes immersed in a water trough in a trough of water. Now allow to stand for the required time, which is actually 24 hours. Then watch the upward movement into the soil samples and record your observation. So this is how you're going to present the method used. Now let's look at the observation. Now, at the early stage of the rise of water in the different tubes, it is observed that the rise of water, which is capillary action, is faster in sandy soil than in clay and loamy soil. So this is how you present your observation. Then you also say, but after many hours or at the end of the experiment, clay and loamy soil experienced higher rise in water, but lower rise in sandy soil. So that's how you're going to present your observation. Now, what are the precautions you should also take note of, or if you're asked to present or mention some precautions you should guard against while carrying out the experiment. Now, soil samples should be dried under the sun. Loam and clay soils should be ground before use. The capillary tubes should be properly plugged with cotton wool. The capillary tubes should be properly tapped to close all air spaces. The capillary tubes should be suspended in a water trough or beakers. So these are the precautions. You could also be asked the reason for the difference in the level of water rise in specimen E and F. Remember, the difference of level in capillary action or water rise in specimen E and F, as was demonstrated or observed in the experiment, is as follows. The difference in level of water in E and F is because F, which is the clay soil, has very fine pore spaces, which allows water to move upwards because of strong capillary attraction, while in sample E, which is sandy soil, There are large pore spaces or large pores with low capillary attraction. Hence, upward movement of water is slow. So this is the reason why there was a faster or higher rate of capillary action in the clay soil as compared to that of the sandy soil. Now we're going to look at another specimen called the Mazeweel. So the Mazeweel is specimen H, and we need to know the botanical name of the Mazeweel. Now the botanical name of Mazeweel is called Cytophilus zmaes. Now take note of the spelling. Remember, the first name is the generic name, and the second name there, that zmaes, is the specific name. Now Cytophilus, that the S, Cytophilus, S, has to be a capital letter. All other letters should be small letter. That's a rule you need to observe when writing the botanical name of specimens. So Cytophilus, S should be capital letter, then Itophilus, all small letters. Zmaes should also be small letters, Zeameais. And remember to underline these words separately. As you can observe, I underline them separately. So take note of that, so that you do not lose your marks. Now the minimum life cycle of the Mazeweel is 28 days, and the eggs are usually laid in stored cereal grains. There's grains that are stored, the eggs are laid there, or in the field, they could be laid by flying adults. So we're also going to look at a particular variety of maize that is usually not damaged or affected by Mazeweel. We know that this pest actually causes a lot of damage to maize grains. And but of all the varieties of maize grains, there's a particular variety that seems not to be affected by the Mazeweel, and that variety is called the flint maize, Ssce indurata. Ssce indurata. Now it's known for its hard kernel. It's known for its hard kernel. The kernel is the seed, so it's Weevil. The Weevil, which is a boring insect, cannot bore into it. So you could also be asked the part of the plant that is attacked by the Mazeweel, and that's the part of the plant that is usually attacked by the Mazeweel, is called the maize grain. Let's look at the damages that this Mazeweel causes to the maize grain. You could also be asked to mention some of the damages caused by the Mazeweel to the maize grains. Now it bores into the grains of maize and creates holes. It also destroys the embryo and prevents germination. Hence prevents germination. Now the embryo is found in the seed, and it consists of the radicle and the plumule. Now the plumule is what eventually develops to become the shoots. Remember, plants are made of the shoot system and the root system, where we have the parts above the soil and the root system is below the soil. So the plumule develops to become the shoot system, and the radicle develops to become the root system. So this Mazeweel damages the embryo, and once that is done, that seed loses its viability, can no longer germinate. Now it also eats the endosperm, which is the food store, and leads to a reduction in the food value. Then the vents, those are the openings as a result of the boring of this Mazeweel, serves as a site for secondary infection. So diseases can actually get into the maize seed as a result of the feeding habit of this insect or this pest. Now, how can we control the Mazeweel? First, you have to dry grains very well before you store them. You should also harvest early, soon after drying. You understand? Harvest early. Then the grains should be stored in airtight containers, and this is also called hermetic storage. So when you store grains in airtight containers, it's also called hermetic storage. So you can write that as a point to help control Mazeweel. Then apply pesticide to the grains before storage. You should also fumigate storage facilities where the grains are stored. It should be fumigated. That means certain chemicals, pesticides should be sprayed there to get rid of potential weevils. Then you could also use smoke to kill or ward off the weevils. And you could also store the grains in hot dried pepper or using neem extract, which is from a plant, the neem tree. So it can be used as a means of getting rid of the maize weevil. We can also take a look at the structure of the maize weevil to gain some knowledge, because you could be asked some questions from here too. Now, the structure of the maize weevil, said it has a hard exoskeleton which is dark brown in color, and the body is divided into the head, thorax, and the abdomen. Now, it has a chewing mouth part with well-developed mandibles, and these mouth parts are elongated and prolonged. If you see the picture there, you can see the mouth part is elongated and is prolonged into a snout called the rostrum. So that boring part, that the mouth of the maize weevil, is called the rostrum. Now, what's the main? I've said that what's the main function of the rostrum is to bore holes in the grains where the females will now lay their eggs. It also has a pair of jointed, bent, or elbowed antennae attached to the mid-length of the rostrum. If you take a look at the picture, you can see the bent or jointed antennae. There's a jointed, bent, or elbowed antennae, which is attached to the rostrum. Take a good look at the picture, you'll see what I'm talking about. Now, the antennae ends in a knob called the scape. We should also know that from this picture of the anatomy of the maize weevil, you can see the thorax is divided into prothorax, mesothorax, and metathorax. And each segment bears a pair of thick jointed walking legs. Now, the weevil also has two pairs of wings. The forewings are hardened and leathery or thickened to form a elytra, which is a wing cover. And these wings meet in a straight line down the middle of the back. So take a look at this picture and you understand that that description fits what you can see. The hind wings are membranous and are used for flying. When the beetles are not flying, the hind wings are folded under the elytra. The bean beetle is another specimen we're going to take a look at. Now, this is specimen I, the bean beetle. You can also call it the bean weevil. Now, what's the botanical name of the bean beetle is Acanthocelids obtectus. Acanthocelids obtectus, which is the botanical name. Remember the rules for writing the botanical name. You underline the words separately. The first word there is a generic name and the second word is a specific name. So Acanthocelids is the generic name, while obtectus is a specific name. On the line, the first letter, remember, you can see the capital A. The letter A starts with a capital letter and all other letters are small letters. If you miss one letter in the spelling, you have missed it. You've missed your marks. You've lost it. So you have to be careful to memorize the spelling of these botanical names. Now, the bean weevil or seed beetles, they belong to the family called Buccinae, and they belong to the subfamily called Buccinae, and they are placed in the family Chrysomelidae. They belong to the phylum Arthropoda, the class Insecta, and the order Coleoptera. Now, they are classified as boring insects. So, the maize weevil is a boring insect. The bean weevil is also a boring insect. And the part damaged by the crops is the seeds. Now, how can the bean weevil be economically important? When we talk about economically important, we mean both positive importance, the positive aspect or the negative aspect of the specimen in question. But in this case, I don't think there's anything positive about the bean beetle. So just take a look at the economic importance of the bean beetle. One, it destroys the viability of seeds. How is this? By destroying the cotyledon of the seeds. It also reduces the market value of crops or the quality of crops due to the boring activities or the feeding activities. It also increases the farmer's cost of production because the farmer ends up spending more on trying to control this pest. It reduces the quantity of stored produce because it usually reduces these grains to powder. Let's take a look at the life cycle of the bean beetle. It has a short lifespan of about 6 to 8 days after emergence. And after this emergence, mating takes place. The female begins to lay eggs before she dies, and these eggs are attached to the surface of the seed, and the larvae bore directly into the seed where they feed and pupate. That means moving to the next stage. Remember the life cycle, we have the egg, we have the larva, the pupa, and the adults, depending on if it's complete metamorphosis or incomplete metamorphosis. So I said the eggs are attached to the surface of the seed, and the larvae bore directly into the seed where they feed and pupate. Adult beetles emerge through circular holes from about 22 to 28 days after the eggs have been laid. Remember, the eggs are laid in those bored holes, and about 3 to 4 beetles emerge from a single seed. So how do we control insect pests of stored grains? We need to look at that. You could possibly ask these questions. Generally, how to control insect pests of stored grains. Now, concrete floors where you store these grains should be used to block insects' hiding places, and there should also be timely harvesting, if possible, removal of infested grains before storage. So timely harvesting and possible removal of infested grains before storage. And before you store, the grains should be properly dried. The grains should also be stored in well-ventilated places. That the stores should be well-ventilated. And for the roof, you should use waterproof and damp-proof or metal roofs, and not using thatched roofs. So these are some of the ways to control insect pests. There are still other ways we need to mention. Say, do not store the products close to the walls or directly on the floor. Spaces should also be left between the grain bags. The stores should be inspected regularly to detect and control pest infestation. The stores should also be fumigated before storage and after removing old stocks or consignments. And local storage methods such as the use of hot pepper, neem seeds, leaf extracts, neem seed oil, or smoking can be adopted. The next specimen here we're going to look at is a grasshopper. Now we'll start with the botanical name of grasshopper, which is Zonoceros variegatus. So remember the rules for writing botanical name. The generic name, the first letter of the generic name should be capital, then all the other letters, small letters. And for the specific name, all the letters are small letters. So let's look at which plants are damaged by the grasshopper. We have corn or maize, wheat, alfalfa, soybean. These are crops that are damaged by the grasshopper. And the grasshopper is designated as a national pest by the Nigerian Agricultural Advisory Council. Let's look at the life cycle of grasshopper. We said the female digs a small hole first into the soil and lays its eggs. It also covers the hole with a funny fluid. Now the eggs later hatch into the nymph, which feeds voraciously on leaves and crops, or leaves of crops. Then this nymph's molts, that changes gradually about 5 times, That molting is like it's kind of metamorphosizing into the adults, removing its outer covering, and after some time, after that it removes it again. That molting that's what it refers to, removing that outer covering. The nymph does this about 5 times before it becomes an adult grasshopper. Now, these adult grasshoppers and nymphs also defoliate crops. That means they feed on the leafy parts of crops. The grasshopper undergoes incomplete metamorphosis, so it's not like the weevil. So it's from egg to nymph to adult, not egg, pupa, nymph, adult. So it undergoes incomplete metamorphosis. Now let's look at alternate host plants that can be attacked or affected by the grasshopper. Banana, cowpea, garden egg, citrus, cola, oil palm, pineapple. So if you're asked, mention alternate host plants that can be affected by grasshopper, you should mention these crops. Now, it's based on its feeding habit, the grasshopper is classified as a biting and chewing insect pest. Now, the parts of crops damaged by the grasshopper, you could be asked to mention the parts of crops damaged by the grasshopper. So we have the leaf, the young shoots, and also succulent stem. Now let's look at the damages caused by this specimen, grasshopper. It causes defoliation of crops, that's the loss of leaves. Young shoots are also destroyed. Wounds are left on the crops that can serve as vents. Wounds or openings are left on the crops that can serve as vents, which are openings for secondary infections. Now the buds are also destroyed. It reduces the photosynthetic ability of crop plants. Why is this so? Because of defoliation, that's the loss of the leaves. Then it reduces the growth of crop plants. How can this specimen, which is grasshopper, be controlled? It can be controlled by hand picking. It can also be controlled by spraying with insecticides. Cotton stainer is another specimen we're going to take a good look at. And the botanical name for cotton stainer is Ssce species. Ssce species, that's what you're going to write. And what is the damage that the cotton stainer does to the cotton plant? Of course, from the name cotton stainer, you can know that that's the specific crop it actually attacks. But there are still other crops that are attacked by or damaged by the cotton stainer. But let's start with the cotton plant. So you can see it damages the embryo of young cotton or balls. Now, causing the cotton balls to abort or fall prematurely. Now, it can also introduce pathogens, which are disease-causing organisms, especially fungus, into the cotton plants, and this is known as internal ball disease. The lint can also be stained. Now, in the cotton plant, we have the cotton ball. We also have the lint. That's that whitish part of the cotton plant. Now, the activities of the cotton stainer can stain this lint. The lint is usually whitish, but with the activities of this cotton stainer, it now stains it. That causes a discoloration of the lint. It can also reduce the oil content of cotton seeds. Remember, cotton seeds are also oil crops. You can get oil from cotton seeds. So the activities of the cotton stainer can reduce the oil content of cotton seeds and also reduce the viability of the cotton seeds. That's the ability for the seeds to grow. And also reduce the market value because the cotton lints are stained by the activities of these feeding habits of this cotton stainer, which actually leads to fungus attack. Let's take a look at the alternate hosts of host plants that can be affected or attacked by the cotton stainer. That means, from the name I told you, cotton tree is actually the primary plant that is actually attacked by cotton stainer. But we still have other host plants that can be attacked by the cotton stainer. We have kenaf, millet, okra, maize, tomato, cowpea, yam, baobab, guinea corn, silk corn tree, silk cotton tree. These are some of the trees or plants that can be attacked by the cotton stainer. Okay, let's take a look at the life cycle of cotton stainer. Now you can be asked this question to describe the life cycle of cotton stainer. So you start by saying the eggs of the insect hatches after some time, about 6 days into nymphs. Now these nymphs go through 5 stages of development, which is also called molting, before reaching the adult stage. And next you say the whole development process takes about 30 to 50 days. And cotton undergoes incomplete metamorphosis. So from egg to nymph to adult, that's just three phases. Okay, so what are the damages caused by cotton stainer to the alternate host plants or crops? It presses into the mature okra fruit or egg apple. Okay, it's also, the vents can serve as a site for secondary infection. It also prevents good fruit formation in okra and causes premature drop of fruits. How can the cotton stainer be controlled? We also need to take note of this. You could be asked to mention maybe 2 or 3 ways the cotton stainer can be prevented or controlled. Now you could state hand picking. You can also say spraying with insecticides or use of crop rotation or regular weeding. Let's look at this specimen called tilapia fish, which is another specimen that was provided. Now we look at the class of tilapia fish. It's called Osteichthiae. I repeat, osteik TAs. And the method of preserving the tilapia fish from the pond is also another possible question that could be asked. So the methods include hook and line, fishing net, using a fishing net like the scoop net, the gill net, and the seine net, using a fishing basket, using a fishing trap, also harpoon, which is fishing with spears, or you could also drain the ponds. And you could also be asked to mention the byproducts that could be obtained from processing tilapia fish. And these are some of the byproducts: the fish bone, the fish meal, the fish scale, fish oil, and the fish skin. You could also be asked method of preserving the tilapia fish. And these are some of the methods that could be used to preserve the tilapia fish: sun drying, salting, smoking, canning, freezing or refrigeration, oven drying, and frying. The next specimen we'll be looking at is the hay. And here we're going to understand or look at some possible questions that could be asked concerning this specimen. So the hay is specimen M. Now, you could be asked to mention the processes or steps in making hay. So first, you cut the forage before flowering or early maturing or bloom. The forage material is chopped. Then you also, the next is spread the chopped forage or regular turning of the spread forage. Next is to dry dry to reduce the moisture content. This could be done in the field or mechanically using forced hot air in the barn, if it's kept in the barn, during the climate, during raining seasons. So you could say dry to reduce the moisture content. This can be done in the field or in the barn. Next point is dry to maintain its green color. And next is bale or pack, and finally stack or store in a dry place. Let's look at the qualities of hay. You could be asked to outline some qualities of the specimen M, which is hay. So let's go down. What you should say is the leaf proportion must be high. The next is must retain a fairly green color. The forage should be cut before flowering. It should be free from dust and molds. It should be free from dust and mold. It must be free from weeds and poisonous herbs. It must have a low moisture content of about 15 to 25% moisture. The next specimen we have is the fish meal, and this is specimen N. You could be asked the main food nutrients in the fish meal, which are protein and fats and oil. You could also be asked the function of these main nutrients. For protein, protein is for growth and body building, production of hormones and antibodies, repair of damaged tissues, replacement of worn-out body tissues, synthesis of enzymes, hormones, and other body fluids, formation of animal products such as the milk, eggs, wool, fur, feather, etc., formation of gametes. There's a male and female gametes, source of energy. Protein is the source of energy, and also the formation of hemoglobin for transportation of oxygen in the blood. So these are the functions of protein. You could also be asked the functions of fats and oil. Fats and oils for insulation or conservation of body heat. They also act as carriers of fat-soluble vitamins, or they are solvents for vitamin A, D, E, and K. Those are fat-soluble vitamins that are dissolved by fats. They also help to protect organs such as the intestines and the kidneys. They are also a source of energy, and they are for medicinal purposes. They can also provide essential fatty acids. Now, the fish meal, let's look at other, still on fish meal, specimen N. You could be asked to mention other feedstuffs that can be used to replace fish meal, because the fish meal is actually used for compounding animal feed. So you could be asked to mention other feedstuffs that could be used to replace specimen N. So you could use the meat meal or meat and bone meal. You could use the blood meal, maggot meal, shrimp meal or crayfish meal, whole milk or skim milk, earthworm meal, feather meal, poultry eggs or offals, insect meals like the termites, the larva or larvae. So these are other feedstuffs that could be used to replace the fish meal when compounding animal feed. Best of luck. Thank you so much for your time.